Hello everyone, welcome to our course on structural equation modeling and generalized linear mixed effect models in R. And my part of this course is dedicated to structural equation modeling. And I'm looking forward to dive into this very interesting topic. And let's start. First, let me introduce myself. My name is Oksana Buzdehan, and I'm the senior scientist in the theoretical ecology group of Britta Titjen at the Free University Berlin. By training, I'm an ecologist, and my research interests are in the relationship between biodiversity and ecosystem functioning and how these are affected by environmental changes. Also, I'm interested in ecological networks, which capture the trophic relationship among species, the flows of energy in ecosystem, and cycles of nutrients. And I have some experience using SEM in the research projects, and we have a few papers published with using structural equation models. And here you can find the link where you can learn more about our projects. And in case if you are interested to join any of these projects, you are more than welcome. And in this part of the lecture, we will learn about the basics of SEM. We will talk about the transition from regression analysis to SEM. Also, we will talk a little bit about the history of SEM and the place of SEM in the natural sciences. Then we will discuss what is the workflow process and where do we start when we want to use the SEM in the research project. And we will have the first impression of the package Lavan in R, which is used to build and assess SEM models. So let's start by discussing how the SEM differs from the regression analysis. So as you know, by the regression model, we aim to understand whether the response variable is impacted by the predictor. And let's take as an example our project which we did in the Carpathian Mountains, and we studied the grasslands and the effects of grazing on plant diversity on the grasslands. But here it's not only the grazing, but also the effects of bare soil and the elevation of the grasslands. Thus we have the multiple regression here. And the regression captures the univariate relationship. So this is a relationship which will involve one response variable explained by a set of predictors. But in our study, we were not interested in the univariate relationship, but rather in the network of relationship among variables. So here you can see that plant diversity is affected by different predictors, where some of these predictors, like bare soil, for instance, are also affected by other predictors, like grazing. So the question now is, how do we test this network of interactions? And such relationship among variables that involve the simultaneous influences and responses are called multivariate relationships. And the structural equation modeling allows us testing such systems of relationship in a model. When we have such network of relationship between variables, such multivariate relationship, we also usually have the direct and indirect effects in a system. So example here that grazing can directly change the plant diversity by consuming the biomass of plants. Also, it can indirectly affect diversity by changing soil properties, for instance, by bare soil exposure. And knowing such direct and indirect effects is important from scientific point of view because it allows to test different mechanisms in a system, from which pathway, through which pathway the effect comes from. And the benefits of CM is that it allows us to do this. Another very special feature of CM is that it allows to test some theoretical concepts which we cannot measure directly, but which we can approximate using a set of observed variables, of measured parameters. And example here is biodiversity concept, which also named latent variable in the CM. And it's approximated by a number of measured or observed indices. And CEM is one of very few tools in statistics that would allow us to do such type of analysis. And now let's think what the CM stands for. The structural means that our hypothesized model has a specific structure. So this is network of relationship where we specifically know where is the cause and where is effect. And then we take this network of causes and effects and translate to a series of mathematical equations. And these equations can be modeled against our observed data. And this would allow us to support or to deny our hypothesized causes and effects. Also, we can say that CM is not one statistical method or technique. It's rather the modeling framework of many multivariate techniques. And we take the statistical and mathematical tools together with CM principles in order to understand our study systems. And CM allows to study different multiple processes in really complex systems. And these CM tools come from the number of different disciplines. As you can see here, it brings together measurement theory, factor analysis, path analysis, and conventional methods of statistics like correlation and regression. The CM also is a part of graphical modeling, and such graphical representations of CM is called path diagrams, which have special notation. For instance, latent variables or our hidden concepts are shown by ellipses. The observed variables are shown by rectangles. Our error variances or unexplained variances by variables are shown by circles. The causal effects are shown by straight one-headed arrows, and the correlations or covariations among variables are shown by curved double-headed arrows. The explanatory variables in the CEM are called exogenous variables, and they always have arrows directed only out of them. And the response variables are called endogenous. They also can have arrows directed out of them, but always they must be predicted at the same time. Furthermore, each path diagram can be expressed also in the form of equations. And then I would like to recall again that CM can represent a complex networks of relationship among variables. So this complex network can be modeled in the form of the path diagrams, also in the form of equations which correspond to these path diagrams. And in this way, CM addresses this multivariate relationship among variables and allows mechanical understanding, allows to test direct and indirect effects. As you remember, the structure of CEM model implies that we have the causality in our system. And this is a very important issue in science because, as you might remember, the correlation does not imply the causation. This is a famous quote of Ronald Fisher. And when we think about causation, we can say that X causes Y if the manipulation of X leads to the response in Y. But in contrast to that, if Y and X tend to be observed at the same time but not necessarily affect each other, then we would say that they do correlate. And the idea of causality in our systems comes from the scientist, from the knowledge of the researcher who builds the hypothesized model. Let's take the relationship between smoking and lung cancer as an example here. In the 1950s, scientists strongly believed that smoking does not affect lung cancer, but rather they do correlate due to some other causes. And it's ironically that Ronald Fisher, who said this famous quote, also believed that smoking is not a cause for the lung cancer. But as we accumulated more knowledge and as science developed, we now have more evidence that smoking causes lung cancer. So when we would build the model for them, we would assume the causality here. And one more example is that when we stay in traffic, we always can see the ambulance also staying there. And if somebody doesn't know what is going on here, would think that ambulance causes these traffic jams, or would think that ambulance and traffic jams correlate because they are also always tend to appear together. But as we have experience and we know our study system, then we can assume that ambulances are caused by traffic jams because traffic jams produce car accidents. But what is very important is to understand that the SEM is not a method for discovering causes. So we don't use SEM to test whether X affects Y or Y affects X. And the SEM totally relies on the causal assumptions made by the researcher who built the model. And these assumptions come from the previous knowledge, from observations, our research design, prior statistical models, prior experiments, and so on. And the credibility of the SEM model depends totally on the credibility of the causal assumptions that we do when we build our model. And now let's talk about where SEM comes from and what is the place of SEM in the natural sciences. Here is some quite oversimplified development history of the SEM, but at the same time it's quite a complicated figure, but you don't have to know it. I want to show simply here where is the SEM methods comparison to the conventional statistics. And as you can see here, the development of SEM took some methods from the conventional statistics such as correlation, chi-square statistics, likelihood estimation, and others. But the development of SEM started from the path analysis and also was strongly affected by the factor analysis initiated by the work of Spearman. And then the father of the SEM, Joris Koch, he synthesized the path analysis and factor analysis into the methods called LISREAL, which stands for the linear structural relationship. And not so long time ago, the Bayesian analysis tools also are implemented in the SEM. What I want to say here is that many things are still not resolved in the SEM because it's a very new technique and it's developing every day, but it is cutting edge technique which integrates the new developments from different statistical methods. As I mentioned, the SEM is a growing body of knowledge, but the majority of the advances in SEM are happening in the human sciences. And in natural sciences, we also see the increasing interest of using SEM in the recent years. And here I show a few people whom I really respect, who are leaders in adapting SEM to the needs of ecology and evolutionary biology. The first one is Jim Grace. And here you can see the website and some papers and books where he did a great deal of work for adapting SEM for ecological research. Also, John Levchik, he recently created the package in R piecewise SEM, which is a strong advance in the tools of SEM. And here also you can find his book in the GitHub website. Also, Jared Burns collaborates on the SEM package and Lavant package, and here you can also find a lot of useful information on the SEM in ecology. And in addition to the previous slide, here are some books where you also can learn about the SEM. And next, I would like to talk about the workflow process of SEM and where do we start if we want to implement SEM in our research project. And here I show the steps of the SEM process. So first we start from the theory based on which we build our hypothesis. And the theory is the literature or the previous experiments or available data or some logical arguments. And based on that, we build our hypothetical network of relationship between the study variables. And then we use this model and we collect data or we get the data from other people who collect it or from literature, and we confront our hypothetical model using the data. And then this allows us to estimate the parameters for our hypothetical relationship and to assess the model fit. And if model and data are consistent, then we interpret model and connect it back to the theory, to our hypothesis. But in case if model and data are not consistent, then we should consider this not as a failure, but rather as a learning process, because given our data and our model, we can learn about our study system. Furthermore, we can revisit the theory and modify the model, and again to assess it given the data. To understand the SEM process, we will use our previous example on the grassland project. So we start from the theory. Based on the literature and previous data, we hypothesized the relationship among these variables in our study system. So we created the conceptual hypothetical model. And now, next step would be to collect the data in order to confront the model with the data. For this, we established grassland plots across the Carpathian Mountains, and each plot differed in the grazing intensity and elevations. And on each plot, we measured the variables which we have in our hypothesized model. So we measured elevation, cattle number as a grazing intensity, percent of bare soil, and plant species number. And you can see here, this is me doing the sampling during my PhD project. So the next step is to use our sample data and to assess our model. And before we do it, let's first check our data. I suggest that together with the lecture, you try to reproduce this code yourself in the R. So for this, we have the dataset named grassland data CSV file, and you will find it on the blackboard. And when we check the dataset, we will see that there are 4 variables, the same as we have in our hypothetical model. And when we plot the pairwise plots of this data, we can see that there are no any surprises here, so we don't see any non-linearities here. And so we will use the data and test our SEM model using the Lavaan package in R. And in this way, we will get the first impression of this package. First, we have to install the Lavaan package, or to call the library if you already installed it. And the first step is to specify our model structure. We have to take our conceptual, hypothesized model and transfer it into the Lavaan. For this, for each our endogenous variable, our responses, we have to specify separate equation with the predictors for this response. And the specification of the model is similar to what we do in the regression analysis. We use a single tilde here to specify the predictor for our response. But here there are no any data set connected to this model, and we take this set of all equations and put them within the single quotes, as I have shown here. Here I show more operators that are used in Lavaan to specify the model. Also, I recall what we already know on the notations of the path diagrams. So the straight single-headed arrow means regression, and for this we use single tilde. The double-headed curved arrow means covariation or correlation, and we specify it using double tilde. We don't estimate intercepts in SEM, but if we need to do this, we can specify it single tilde 1. The latent variables, which are notated by ellipses, are the theoretical constructs, which are measured by other variables, and we can specify them using the sign equal and single tilde. And the composite variables is a new term for you, and these are similar to the latent variables, but we will learn more about this in our next lectures in this course. And it can be specified as less than sign together with a single tilde. So the package Lavaan stands for the latent variable analysis, and I suggest you revisit the web page for it in order to get more understanding on what is possible in Lavaan. And the next step is to confront our model with the data and to estimate the parameters. So for this, we use function SEM for our specified model. Here it is, SEM mode, and also we define the data with which we want to fit our model. And then we want to extract the result. For this, we use the function summary for our fitted model. And now we will get the warning, the error. So Lavan is saying that it has some problem with our data, and this problem is related to the scale difference of our variables. And this is because Lavan is a bit fuzzy about the difference in data scales because it affects the manipulations of the covariance matrices needed for the maximum likelihood assessment. And this seems to vary from version to version with Lavan, but we don't have to address this problem, but I still want to show you how we do address it. And for this, we can inspect the covariance matrix of our model. For this, we use function LoveInspect for our fitted model, and we use argument co because we want to see the raw scales of the data. And we see here that our variables do differ in the scales. But there are no any missing data, so there is no problem with the data itself. But if you want, if you are worried, we still can rescale data to the same scale. For this, we have to check information about our variables. We can use function varTable for our fitted model, which gives us each variable and the information for them. It even shows us which variable is exogenous here. We look for the mean and for the variance for each variable, and indeed you can see that they strongly differ in their scales. What we have to do, we have to bring them to the roughly similar scales. So for this, I rescaled the diversity and elevation. And now I will repeat the model estimation again using this transformed data. And you will see that this transformation solved our problem and we don't have any more the warning. And if you compare these results to the previous results, you also will notice that there are no difference in the results. This means that we don't have to do these transformations in the future. And the first part of the summary output will give you the assessment of model fit. And we will talk about this in the part 3 of the lecture today. If you read further through the output table, you will notice the results for the regressions. The first column here shows the raw unstandardized regression coefficients for each relationship. And standardized errors are simply the standard errors for these estimates. And the third column are the z-values calculated from the Wald statistics. And P are the significance levels for our estimates, for our parameters. If you specify argument standardized equal true in the summary function for our fitted model, then it also gives us the two additional columns. And the first column shows the standardized coefficients for latent variables, while the last column will give the standardized coefficients for all variables. So we don't have here the latent variables, so it should be the same as the raw estimates. But here I highlighted in red and blue the raw and the standardized all regression coefficients. There are also some tools to visualize the path diagrams in Lavan, but I just want to specify here that these tools are not so sophisticated as you know for the regression visualization, so it's quite limited. For this, we can use library Lavan plot, and we can plot our model. So we can use the same function, Lavan plot, and specify our fitted model. And also, if we want our coefficients to be shown in the path diagram, we specify argument coefficients equal true. And then you will have this raw unstandardized coefficients exposed here. And if you want standardized coefficients, then you should specify argument stand equals true, and you will get the standardized ones. And this is important because in many studies we use the standardized coefficients in order to be able to compare the effects across different pathways, across different links in a model. If you want your graph look more like a circle, not like a hierarchical model, then you can change layout here using argument layout equals circle. Also, sometimes we don't want to see the non-significant coefficients, for instance, like here. And that's why in your graph, you can specify argument zik equal 0.05. But as I mentioned that this visualization functions in R for the SEM models are quite limited. For instance, you can see here the overlap of arrow and the path coefficient. And I usually use these functions to check the structure of the model and to check the results. But for the final visualization, I use some other not R tools. But you can check more about this package and maybe you will find it really helpful or interesting. Another package that is really widely used in the humanities sciences is SEM plot. I have never used it because I found it hard to read, but for this you can use the function SEM path, where also you can change different parameters for your plot. And more information you can find here under this link. And this was the basics of structural equation modeling. And in the next part of the lecture for today, we will learn about the path coefficients. So looking forward to see you then.